And on this slide, you can see the growing of the antibiotic resistance bacteria and the result of antimicrobial activity. According to the compounds, according to the determination of composition of time essential oils, we see that these essential oils contain the highest level of thymol, yes, 67%. The determination of antibiofilm activity showed that the highest effect was characterized to the Thym essential oils and Oreganum essential oils, and we investigate the influence of essential oils on biofilm for prevention biofilm formation, and also we measure the influence of essential oils on already formed biofilm. The highest antibiofilm activity we have shown for Thymus and Oregano essential oils, and on the picture also you can see the effect of different concentration of Thymus vulgaris essential oils and Oregano vulgaris on the already formed biofilms of Staphylococcus aureus. Then the next step of our investigation was the antimicrobial and antibiofilm effect of the plant extracts, and we divide all plant extracts which we investigate to the several group with the high antimicrobial and antibiofilm effect. It was the ethyl leaves extract of Vaccinium vitis idea, it is a red berry, and Potentilla erecta. Then the extracts which had only antimicrobial effect. It was fruit of Vaccinium vitis idea and the flowers of Arnica montana. Then the group with antibiofilm and weak antimicrobial effect, and some of the species has only antibiofilm activity, and group of the extracts with which hasn't any antimicrobial and antibiofilm effect. So the highest effect to the highest antimicrobial and antibiofilm effect was shown for the ethyl leaves extract of Vaccinium vitis idea. The results you can see on the table, and this picture on this picture you can see the antibiofilm activity of the different concentration of ethyl extract of the leaves of Vaccinium vitis idea. Also, the high antibiofilm effect we observed for the extracts of Potentilla erecta and for the Equisetum arvense. The next step was to create some composition from the essential oils and extracts which have additional antimicrobial and antibiofilm effect. And as a result, we make some composition with high antimicrobial and antibiofilm effect, and we developed a series of medicinal and hygienic products for the care of the oral cavity. And now these solutions at the stage of before clinical research on the basis of the University Dental Polyclinic. On this diagram, you can see the antibiofilm activity of this oral care solution and the results of the antimicrobial and antibiofilm activity of the oral care products. So, thank you for your attention.
Thank you, Professor KRYVTSOVA, for very interesting information concerning natural weapons against so important problem as antibiotic resistance. And we already have some questions in the chat. Let me check. Sorry, it's not the first. And the first question is, how you differentiate antibacterial from antibiofilm activities?
We measure the antibiofilm activity with the methodics in I show on the slide in 96 wall plate. At the first step, we create, we make this experiment in two variants. We put the essential oils and plant extracts before biofilm formation, and the second variant of experiment it is to put these extracts and essential oils on creating on formed biofilm. If the extracts and essential oils hasn't showed antibiofilm effect, we think that it is only antimicrobial effect. If we have only antibiofilm effect but hasn't antimicrobial, we select these plant extracts to the group with only antibiofilm activity. So if we also measure the minimal inhibit concentration of each plant extract and essential oils, and our conclusion, if we have and antimicrobial and antibiofilm, it was the two effects of some plant-based origin substances.
Thank you. And our next question is, thank you for the speech. Have you tested the possibility of developing allergic reactions, and what reference percentage of possible allergy development cases is reported in the literature?
You know that allergic reaction is an individual property of organisms. Yes, it is possible. Yes, it is possible, but for all medicinal plants which we use, it is now this plants and extracts and oils is wide used in cosmetics, in a different parts of our life. But all components, all things may make some allergic reaction. We know about this.
And the last questions for this speech. Have you studied the influence of plant compositions on normal microbiota representatives? Are there any changes in the composition of the normal microbiota of skin or mucous membrane of the nasal cavity and possibly the gastrointestinal tract?
Yes, we measure these compounds on some lactobacillus strains, and we have a low effect on these strains, but I should note that these compositions and compounds not for the health oral cavity. It is only for oral cavity with some disorders in microbiome, and especially with the chronic inflammatory processes such as periodontitis disease. So it is not for the regular use for the normal microbiota. Thank you. Thank you very much for your question.
Thank you again for your amazing presentation, and research area of our next plenary